Hi guys, this is Sahar, and today I'll be doing chapter 31, which is on animal origins and the evolution of body plans. So today we'll only be going up to 31.3, and the next podcast will finish up the chapter. Alright, so here's the table of contents. So as I said, we'll be going up to 31.3 today. So 31.1 will be covering what evidence indicates that animals are monophyletic. Then we'll focus on what are the features of animal body plans. And then we'll finish up with how do animals get their food. Alright. I've noticed that often a lot of people ask, like, how much of each section should you know for the exam? So I had an AP Bio prep book, and in that there was a list of, like, kind of a rough estimate of how much of each topic you should know for the AP Bio test. So I kind of took the parts that were pertaining to this chapter, and I've put it into a list here. So hereditary and evolution is 25% of the test, and 8% is evolutionary biology. So that would be early evolution, evidence of evolution, and mechanism of evolution. So we're kind of going to focus on early evolution and evidence of evolution. 50% of the test, which is like a pretty big chunk, is going to be in organisms and populations, and 8% is diversity of organisms. So that includes evolutionary patterns and evolutionary relationships, and we're going to touch on both of them. And 32% is structure and function of plants and animals, and that's probably what we're going to touch on the most in this chapter. So reproduction, growth, and development. Structural, psychological, and behavioral adaptations and response to the environment. So not so much response to the environment, but all of these we're going to focus on. Alright, so let's get started. So 31.1 is what evidence indicates that animals are monophyletic. So what does monophyletic mean? So kind of take a second to try and remember it from the previous chapters. So monophyletic means referring to a group that consists of an ancestor and all of its descendants. Now that we've got that out of the way, let's move on to what traits distinguish the animals from other group of organisms. So all animals are multicellular. That means that they develop from a single cell into a multicellular mature adult. So basically this is saying that animals have many different life cycles and that consist of complex patterns. And that means that they develop from a single-celled zygote into a multicellular adult. All animals are also heterotrophs. That means that they are able to synthesize organic molecules from inorganic molecules. So basically this means that we feed on things, not that they photosynthesize their food. Animals use internal processes to break down material from the environment and organic molecules. So most animals ingest their food, like we just said, into an internal gut, so that would be our digestive system, which is continuous with the outside environment. And most animals can move. Now animals have specialized muscle tissues that allow them to move. Now either they move to get their food or they move their environment to bring the food to them. And many animal body plans are specialized just for movement. Alright, so animal monophyly is supported by gene sequences and morphology. Now the biggest evidence that all organisms considered to be animals share one common ancestor is that they share derived molecular and morphological traits. So, for example, it could be gene sequences such as rRNA, similarities in the organization and the function of the Hox gene, unique type of junctions between their cells, like tight junctions, desmosomes, gap junctions, a common set of extracellular matrix molecules, such as collagen and proteoglycan. So right here, you should already know what Hox genes are and these junctions are and proteoglycans. If you don't, just take a second to go back and review them before you move on. Alright. So the traits that were mentioned before are synapomorphies. Now, what are synapomorphies again? They are traits that arose in the ancestor of a phylogenetic group and are present in all of its members. And this is what helps define a group. So the ancestor of animals was probably a colonial flagellated protist, and it is similar. We think that it's similar to an existing colonial choanoflagellate. Now, here's a theory on how animals came about from a colony of choanoflagellates. So first, the cells in the colony of the protist started to specialize. So for example, they would specialize in movement, some in nutrition, some in reproduction, etc. And this is known as functional specialization. And as functional specializations in these cells continued to differentiate, we got two things. So there was coordination was improved by specific regulatory molecules, and these coordinated groups eventually evolved into larger and more complex organisms, which we now know as animals. So synapomorphies indicate animal monophyly and cannot be used in finding evolutionary relationships because all animals have them. So basically this is saying that synapomorphies show that all animals are related, but we don't know how they evolved from the common ancestor. So to find that, we need to use evolutionary relationships, and these are sought through derived traits that are in some groups but not the others. And these can be found in fossils, patterns of embryonic development, morphology and physiology of living animals, structure of animal modules, and genomes of animals. So here is a tree of all the animals, well, not all the animals, but these are groups of animals that have come from a common ancestor. So this phylogenetic tree shows an interpretation that is based primarily on molecular data. So as we've learned before, phylogenetic trees can be classified by different characteristics and behavior, or different habitats, and so forth. But this is strictly on molecular data. And the traits that are highlighted in the red circles, so these traits right here, we will be reviewing later in the chapter. Alright, so developmental patterns show evolutionary relationships among animals. Different patterns of embryonic development traditionally provide some of the most important clues to animal phylogeny. So I'll be showing a picture of what I mean by this in the next slide. So the first few cell divisions of the zygote are known as the cleavage. And a number of different cleavage patterns exist among the animals. So this is really early development of a animal. So here's a picture of embryonic development. So this is for a fish. This one is a salamander, this one is a turtle, a chicken, a pig, a dog, a monkey, and a human. So if you look at the really early embryonic stages, they look fairly similar, like really, really similar. Then as they start to develop, they start to kind of differentiate. So these two look similar, these two look similar, these two look similar, and these two look similar. And then as they develop and mature even more, then you start to see them really differentiate. So these early stages of embryonic development is really what show us the evolutionary pattern. So it's showing that we had one common ancestor, and that we are then differentiating and then, splitting apart at the nodes as we had seen in the phylogenetic tree, and then differentiating into different animals, different groups of animals. So now back to the cleavage. So cleavage patterns are influenced by the configuration of the yolk. So what is the yolk? The yolk is the mutual material that nourishes the growing embryo. So it's kind of like a little mini food sack for the embryo. So reptiles, for example, have an incomplete cleavage. So this is a type of cleavage. And it means that they have cells that form the embryo on top of the yolk mass. So here's a picture from top view of a chicken embryo. So you can see this is the yolk around, and this would be your mass of cells. And as they start to divide, so this is your cell stages, it does not penetrate through the whole yolk. You just kind of get your mass just kind of builds up on top of your little ball of food. So here's a picture from the side view. So this would be, these little globular pieces are going to be your yolk, and these are your cells that are starting to develop on top of your yolk mass. On the other hand, there's also complete cleavage. So when small yolk particles are evenly distributed throughout the egg cytoplasm, this cleavage is considered as a complete cleavage. So fertilized egg cells divide in an even pattern, which is known as radical cleavage. So radical cleavage is a type of complete cleavage. And this is seen in echinoderms, such as sea urchins. So radical cleavage comes from an ancestor known as eumetozoans. And it is also found in some protozoans and diploblastic animals such as deuterozoans. And we'll be talking about these groups of animals later on. So here's a picture of the two types of complete cleavages. So both of these are complete cleavage, but the first one is also a radical cleavage. Since as you can see, it becomes like symmetrical and it's all around, and it's coming from technically a center point. Now this one, you can see the little yolk particles are distributed around, and then the cleavage forms, and you get the radical cleavage at the blastomeres. Now this is a embryo of a frog, and this is still considered a complete cleavage, even though it's not symmetrically dividing the embryo in halves. In this, the yolk mass sits at one end, and that's known as the vegetal pole, and the top is the animal pole. So that's kind of something like we learned before. And the only, even though it does not have yolk particles that are evenly distributed across the embryo, it's still considered a complete cleavage because the cleavage penetrates throughout the whole embryo. It's not just the top part, as we had seen in the incomplete cleavage. You also get something called a spiral cleavage, which is a complicated derived permutation of a radical cleavage, and it is found among, I'm going to butcher this, but euphotrichozoans. So euphotrichozoans are things such as earthworms and clams, and spiral cleavage, or the animals that have spiral cleavage, are also known as spiralians. So here's a picture. It is kind of complicated. So basically you have a cell and then it starts to divide, but it doesn't just evenly divide and stack on top of each other. They start to kind of twist around and around until you kind of get this like spiraling formation. Alright, so during early development, distinct layers of cells also form, and each differentiate into specific organs and organ systems. So you have diploblastics, which have 2 layers, and triploblastic, which have 3 layers. Now the diploblastic have the ectoderm and the endoderm, which are the outer layers and the inner layers. And the triploblastic have ectoderm, then the mesoderm, and then the endoderm. So it's outer, middle, and then the inner. So now we're going to move on to something called gastrulation. So this is in early development a hollow ball about like 1 cell thick. It's indents and it forms like a cup-shaped structure. And the opening of this cavity is called a blastospore. So here's a picture, and so this would be your blastopore, and this is kind of like if you cut it, but this is basically what we're talking about with the gastrulation. So the development after the blastopore divides the triploblastic group into protozoans and deuterostomes. Can never pronounce that. Anyways, so the triploblastic, as we had talked, have 3 layers, and that group can be divided into these 2 groups. So protozoans are ones that means Greek for mouth first, and that's because the mouth arises from the blastospore, and then the anus comes later. And then we have deuterostomes, which means mouth second. And this is because the anus comes first from the blastospore, and then the mouth comes later. And this was the ancestral condition. So this is like what our ancestors used to have. So here's a recap for 31.1. So animals are thought to be monophyletic because they share many derived traits, such as multicellularity, mobility, and heterotrophic lifestyle based on the ingestion of the outside nutrition. Evolutionary relationships among animals are inferred from fossils and from molecular and mental traits that are shared by different groups of animals. Alright, so now chapter 31.2, which is what are the features of animal body plans. So a body plan is the general structure of an animal, the arrangement of the organ systems, and the integration of its parts. So there are 4 key features to this: the symmetry of the body, the structure of the body cavity, the segmentation of the body, and the external appendages that move around the body. So these affect the movement and the interaction of an organism with its environment. And since animals share common regulatory genes that determine development of body symmetry, cavities, segmentation, appendages, many animals share body plans too. So most animals are symmetrical. So the overall shape of an animal can be described by symmetry. So symmetrical is an animal that can be divided along at least one plane into similar halves. Otherwise, it's considered asymmetrical. So that means there are three types of symmetry. There's spherical symmetry, radial symmetry, and bilateral symmetry. So spherical symmetry is that body parts radiate out from a center point. Just like a sphere, you have all these points that are a certain radius away from the center point. So they have infinite number of planes for symmetry. They are widespread among unicellular protists, and most animals possess other forms of this symmetry. So most animals do not have spherical symmetry. The next type is radial symmetry. So that means that there's one main axis around which the body parts are arranged. So there's also something called perfect radial symmetry. So that means that the organism can be divided into symmetrical halves by any plane that consists of the main axis. So now most radially symmetrical animals are slightly modified as adult. So that means that fewer planes can divide them into identical halves. So maybe when a certain organism is younger and it's smaller, it has more planes that can divide it into symmetrical halves, but as it grows older, it has fewer planes that divide it into identical halves. Now, many animals that have radial symmetry are sessile, and sessile means that they cannot move. So here is a picture of radial symmetry. So this area right here is your little point, and these are all the planes that are coming out, and this is your main axis, sorry, not your point. It would be your main axis, so it would run down through the organism. So this is a top view, basically. And these would be, for example, some of your planes that can divide the organism into symmetrical halves. The other type is bilateral symmetry, which is a characteristic of animals that move in one direction. For example, fish. So animals can be divided into mirror image, left and right. And they have a single plane that passes through their midline. So for example, a fish would be kind of like if you followed its spine. So the midline connects the anterior of the body to its tail, which is the posterior. And if a plane is created perpendicular to the midline, you should get two dissimilar sides. Basically, if you take a fish that's facing you and you make it so that it's like facing right or left, and you cut it in half, you would have like a tail end and a head end, and those are not symmetrical. So the top or the top would be the dorsal surface, and the bottom would be the ventral surface. So basically, if you took a fish and you sliced it in half horizontally, the top would be your dorsal surface and the bottom would be your ventral surface. So here would be your fish, and this is how you would get the mirror image. So that's from the front view. So this would be your right and your left, and they are symmetrical. But if you cut it this way, you would have a dorsal surface and a ventral surface. Alright, so bilateral symmetry is correlated with cephalization, and this is the concentration of sensory organs and nervous tissues at the anterior or the head of an animal. So if you think about any animal that moves in one direction, so for example, a fish, all the sensory organs are at the anterior, so at the face you have eyes, you have the mouth, and so forth. And for us, we have our eyes, our nose, our mouth, our ears. They're all at the front. They're not facing the back. So it has been evolutionary favored since the anterior typically encounters the new environment first. So a fish swimming into, let's say, hot water would first face it with its anterior. So it makes sense that all of its sensory organs should be at the anterior. Alright, we're going to take a quick break, and we're going to watch a completely irrelevant video for bio so that you can kind of relax your brain, and then we'll go back. So, enjoy. Alright, so now back to work. Break time's over. So the structure of the body cavity also influences movement. And there are three types of body cavities, and they're based on the internal fluid that fills the body cavity. So there's achelomate, pseudolomate, and coelomate. Alright, so the achelomates lack an enclosed fluid-filled body cavity, and instead they have the space between their gut and their muscular body wall is filled with masses of cell called a mesenchyma, and they typically move by beating cilia. The other two body cavities, they both are between the ectoderm and the endoderm, and they're differentiated by their relationship with the mesoderm. So now this is what the achelomate would look like. So there's no fluid in here anywhere. So this would be your muscular layer, this would be your ectoderm, and this would be your gut, and this would be your mesenchyma. So these are your masses of cell that filled your body cavity. So the pseudocoelomates are a body cavity and is a pseudocoel. So they have a fluid-filled space in which organs are suspended. So the muscles, which is the mesoderms, are only on the outside of this fluid-filled space. And coelomates, which have a body cavity is a coelom, is lined with a layer of muscle tissues called the peritoneum, which also covers the organs. So they have more control over movements of fluid in the body cavity. So here's a picture of a pseudocoelomate. So this right here is your endoderm, this dark blue ring, and then this red is your muscle, so your mesoderm, and then these yellow would be your gut, so that would be any of your like internal, and then these would be like your internal organs, and all this space would be all fluid that would be around it. On the other hand, you would have your coelomates, which would have your dark blue ring, which is again your ectoderm, and then this dark pinkish reddish ring is your muscle, and then this light pink would actually be your peritoneum, which is again muscle that surrounds your internal organs, and your gut, which is right here. And this would be fluids filled right here, all these empty spaces. Okay, so body cavities can act as hydrostatic skeletons. So that means that fluids are incompressible. So if you have hydrostatic skeletons, your fluids are incompressible. So that means that when muscles contract, it pushes fluid to another part of the cavity, and this causes that region to expand. And this allows for movement of specific body parts. So if you've ever played with like a water balloon and kind of just squished it around for a while, if you squeeze one end and you still have space on the other end, you'll notice that the water will go to the other side and that part will kind of blow up a little bit more than the other. So that's basically what this is talking about. So for example, a snail coming out of a shell. So snails have a hydrostatic skeleton that would not be including their shell, of course, and a snail, for example, is an animal that has both circular and longitudinal muscles, and this allows them to have a greater control over movement. So here are some examples of hydrostatic skeletons. Sorry, let me rephrase that. Here are some examples of organisms that have hydrostatic skeletons. So, for example, jellyfish, starfish, and even caterpillars. They also have hydrostatic skeletons. So this does not mean that you can go around and like completely squish a caterpillar and nothing will happen to it. But it's that the fluids can easily kind of move around through the organism's body. Just like a water balloon. If you push it too much, it will pop. But it does have some flexibility of where the fluid can be. Alright, so segmentation improves the control of movement. So segmentation facilitates specialization of different body organisms or body regions. So segmentation cannot always be seen, so it could be internal. Also, it allows animals to alter body shape and control movements precisely. Segmentation evolved independently several times before it is what we know as now. So the radiation of the arthropods was based on the changes in a segmented body plan. So the legs of a spider are all different segments of the spider's body plan. And these appendages enhance locomotion. So locomotion is important for finding food. So it's important for a animal to go out so that they can capture or get their food. It's also important for finding mates and also important for avoiding predators. So many echinoderms have tube feet, and others will have limbs which are highly specialized for rapid and controlled movement. So we have limbs, so our legs, our arms, they allow for rapid movement, but they also allow for controlled movement. So arthropods and vertebrates have jointed limbs. So those are limbs that are joint, and they are also used for rapid movement and controlled movement, but they're kind of like a subset of limbs. And this is highly important for their evolutionary success. And many of these limbs were later modified into wings. So here's a recap of 31.2. So the body plans of animals are all variations on patterns of either symmetry, body cavities, and segmentation, and appendages. All four can have bearing on movement and locomotion, so which are important aspects of the animal way of life. Alright, so let's get started. So while many animals, such as heterotrophs, which are other feeders, rely on photosynthetic endosymbiosis for nutrition, most animals have to actively obtain nutrition from the outside sources, which is also known as food. So the need to locate food has been favored the evolution sensory structures that can help the organism find food. So such structures could be that they give detailed information about the environment, a nervous system that can receive, process, and coordinate that information that is given by the environment. And so to acquire food, energy must be used. So this is done by moving to the source of food, or to move the environment and the food it contains. So basically, animals can either go and capture their food, or they can bring the food to them. And this leads to two different types of organisms: motile and sessile. So motile is animals that can move from one place to another, and sessile are animals that are stationary. So motile organisms go and get their food, and sessile organisms have to change their environment to bring the food to them. So here are some feeding strategies. So there's filter feeders, herbivores, predators, parasites, and detritivores. And detritivores, you might have heard them as decomposers. So filter feeders capture small organisms delivered to them by the environment. So they use straining devices to filter out small organisms and organic molecules from the air or the water. So many sessile aquatic animals rely on water currents to bring the food to them. But filter feeders don't have to be sessile. For example, I believe flamingos are also filter feeders. But there are aquatic animals that are sessile and rely on their environment to bring them food. Motile filter feeders bring the food-containing medium to them. On the left, you have sessile marine animals, which are also filter feeders. And this one allows the ocean currents to bring it food, and they filter out the food from the water. And on the right, we have motile filter feeders, such as the flamingos, and they use their legs to stir up the mud at the bottom of either the ocean or the lake that they're feeding on, and then they strain out the organisms that they need from the muddy mixture with their beaks. So the motile part of this is, first of all, that they can move, and that they stir their environment to get the organisms, while on the left, these sessile aquatic feeders, they rely on the ocean current to stir the water around them in order to get their food. So some sessile filter feeders have to use energy to move the water past them. So they might not have ocean currents all the time, and so they have to themselves move the water around them. So sponges beat their flagella of specialized cells called choanocytes to achieve this. And these cells link animals with choanoflagellate protists and fungi into a clade called opisthocanths. So here's an example of what a sponge would be doing in order to move the water around it. So you can see how the water would come in through these pores. So the water and the food particles come in through the pores into what's inside this area would be a water canal. And over there the food is filtered and is absorbed into the organism, while the rest of the water is dispelled through the osculum throughout here. And so here would be your choanocytes, and here's your flagellum. So they keep on kind of vibrating, and then this moves this sponge kind of a little right, left, a little bit, and then the water is kind of pumped through it almost. So herbivores eat plants. So they either eat plants or part of plants, but they often don't kill the plant. So that's one really important piece. That herbivores don't kill plants, they eat plants. And many kind of herbivores may feed on a single plant. That means that you can have a caterpillar, a butterfly, a hummingbird, and a lot of different organisms that could be feeding on one plant or in one area. And so that means that they do not need to expend their energy killing or subduing a plant, but they do need energy for digestion. On the other hand, you have like predators and other organisms that need energy to just find and subdue their prey in order to eat them, along with using energy for digestion. So land plants have tissues that are difficult to digest and chemicals that must be detoxified. So that's where most of the energy goes, into digestion of plants. So therefore, herbivores tend to have a long, complex gut for digestion of plant materials. So if you've heard of the cows that have like seven stomachs, they use that to kind of eat and then digest and kind of regurgitate and then eat and digest. It's because there's so much to break down chemically in order for that plant and the for the plants that they eat to be digested. So predators capture and subdue large prey. So when they capture and eat other animals, they're typically larger than themselves. So predators have structures such as sharp teeth and claws and well-developed sensory organs to detect prey. So one of the examples of a sensory organ that is used to detect prey is the two eyes in front of our face. So like for example, we are predators, and these help us gauge distance. And that's one of the major sensory organs that helps us basically be a predator. So predators may pursue or stalk their prey, and they're often in camouflages, and they also use venom as a weapon to subdue large prey. For example, cnidarians have toxins in specialized stinging cells that contain stinging organelles called nematocytes. And these inject toxins into the prey and therefore subduing them, and then they can consume them. And for a real quick thought, cnidarians are a type of jellyfish. Yeah. So omnivores, all you need to know about that is that they're animals that eat both plants and meat. And what they eat can differ throughout their life. So for example, they could be songbirds that eat insects when they're young, but when they grow older and become adults, they usually eat fruit and seeds. Parasites live in or on another organism. So the organism which they live on, they obtain the energy and nutrients from them. So the organism that they live on is called the host. And they're usually smaller than the host, and they do not kill the host. So just like herbivores don't kill the plants, they use part of the plants, or eat part of the plants. Parasites don't kill the host, usually. They usually are just taking a portion of their nutrients. So first they must overcome the host defense system, which is one major thing for a parasite. And so to do that, they usually have complex life cycles, which can contain like dormant stages and then like awakening stages in order to get through the defense system of an organism or be passed through organisms so that they can get to their intended organism. So there are two types of parasites. There are endoparasites and ectoparasites. So endoparasites live inside the host, and ecto live on the outside of the host. Now, by now, I believe you guys have started to notice this little pattern of endo and ecto. Whenever you see endo blank, it's usually inside, and ecto blank is usually outside. So endoparasites are morphologically simple. They often have no digestive system. They just kind of absorb the nutrients directly from the host, so flatworms. Ectoparasites are more morphologically complex, and they often have mouth parts to pierce or suck host fluids and digestive tracts, such as fleas and ticks and leeches. So now we're done with chapter 31.3, and so here's a real quick recap. So animals have many ways of acquiring food, and filter feeders strain food particles from the water or air. Herbivores have digestive adaptations that allow them to eat plants. Predators are physically adapted to capture and subdue other animals, which are prey, and consume them. And parasites obtain their nutrition from a host organism. So that's all for this podcast, and thank you guys for watching. And I'm going to end with a funny video to relax everyone's minds, and hope you enjoy.